In this video, we're going to explore neural development. So this is, again, part of our option A for IB, higher level biology, our neurobiology and behavior units. We have three germ layers that are developed early in development. So the embryo forms three distinct what are called germ layers, and each of those germ layers will further develop to give rise to various tissues and organs. So remember, right after fertilization, the zygote keeps dividing without specializing. So all of those cells are totipotent stem cells. They can become any type of cell in the body. But early on, even in early stages of development, those cells first develop into two layers of cell types and then three. And those three layers now aren't totipotent, they're pluripotent. So they will be able to become only a certain type of cell, like of a collection. So the first layer is the endoderm, that's the internal layer. And the types of cells that are formed from there would be any lung cells, thyroid cells, your digestive cells, so like your digestive organs and so on. The mesoderm is the middle layer, and it gives rise to muscles, so cardiac muscle, skeletal muscle, and smooth muscle. And then also the tubule cells of the kidney and red blood cells. And then finally, the ectoderm, the external layer, would be your skin cells, so your epidermis, your neurons, and your pigment cells. So those are the three germ layers. The first step is neurulation, so chordates. And chordates are organisms that have a spinal cord, so us and other animals with a spinal cord. They develop a dorsal nerve cord, so along the back of the embryo, early in fetal development. And in humans, that actually occurs in the first month of pregnancy. So you can see here the three layers. So we've got our ectoderm, the middle is the mesoderm, and then the endoderm. And this will be the gut cavity eventually. So the first step that happens is an area of the ectoderm, so right here, develops into what's called the neural plate. That neural plate, the cells change shape, and the plate will start to curve inwards into a groove. And then the next step will be the lateral edges fold together, so the two edges right here and here will fold together to form a neural tube. So this is going to give rise to our spinal cord. And then you can see that the ectoderm then closes over that again. And then step 4, the neural tube elongates as the embryo develops. And the hole in the center of the tube right here remains as a canal in the center of the spinal cord. So one of the skills for biology for the option is to demonstrate the neurulation in xenopus. So that's an animal. And you can see this is going to be a dorsal fin, so it's an aquatic animal. You're to copy the diagram, and you need to be able to annotate to show the following. So you need to show the three germ layers, the ecto, the endo, and the meso. So this would be the ectoderm, the mesoderm, and the endoderm. And then the development of the neural tube. So this is our neural tube right here, the red one. The wall of the developing gut cavity, and the notochord, and the developing dorsal fin. So that's the dorsal fin. An application you have to discuss is the condition known as spina bifida. So if the neural tube fails to completely close in a region of the spinal column, that condition is called spina bifida. So there's no bony vertebrae that will form around that section, so the spinal cord will be exposed. The most common site for spina bifida is in the lower spine, and it can cause paralysis in a baby if it's born with its spinal cord exposed. Another thing you need to be able to discuss is the effect of strokes, and how they're caused. So a stroke happens when the blood supply to the brain is interrupted. And because the brain's so active, it needs a constant supply of oxygen and energy. So if it's not getting that, that's a problem. So if a blood supply to a portion of the brain is cut off, the neurons surrounding that area will be unable to perform cellular respiration. If you can't perform cellular respiration, you die. And the effects of a stroke can range from unnoticeable to fatal, depending on how much of the brain is affected and where in the brain the stroke takes place. Neuron development. So once the neural tube is formed, the cells are continuing to specialize. And they'll specialize into the cells that make up the central nervous system. Some of the cells will become neurons, which proliferate to make up the billions of cells of the brain and the spinal cord. And then cell division will stop in most of those parts before birth, but there always will remain some neurons in an adult brain that do maintain the ability to divide. So this is newer knowledge than before. It used to be thought that once you develop your neurons in childhood, you can't create more, but that's not true. There are still some cells that maintain that ability to divide. Once the neurons have developed, they migrate to take up residence where they're going to exist. So they actually move by contractile actin filaments within the cells. So the neurons actually physically migrate to where they need to reside. At this point, the cells did not have axons, so the next step would be to develop axons, and a chemical stimulus will cause the neuron to send out a projection that will become the axon. Only one axon forms per neuron, but the axon could be highly branching. And there's going to be lots of dendrites forming along the cell body to be able to synapse with other neurons to bring impulses from other cells. Then the axons grow. Just like a plant, the axons grow at their tips. They don't grow elsewhere. Some of them will remain short and they'll form connections with other neurons in the central nervous system within the brain. They don't need to have long axons. Others will extend outside of the central nervous system to go connect with muscles or organs. And then some axons in humans can be over a meter long. So that's pretty mind-boggling in itself. And if the cell body remains intact, a severed axon can regrow. So that's an interesting fact as well. So the axons and dendrites will grow until they reach a cell they can interact with. And then once they get close to one another, they will develop a synapse with that other cell. And each neuron can have anywhere from 2 to hundreds of synapses with other cells and other neurons. The next step will be the elimination of synapses. So we can actually form new synapses at any point in our life. So the adage, you can't teach an old dog new tricks, is not true. You can form new connections all the time. We're constantly learning about our environment and we're constantly storing memories. Every time a signal travels across a synapse, chemical markers will be left to strengthen that connection and that synapse. But if a synapse doesn't get used, it'll eventually be eliminated because there's no chemical markers left behind. So synapses really do work on the use it or lose it principle. So rehearsing something over and over again really will make you better at remembering the task, like when you study for a test. You have to leave chemical markers behind by rehearsing over and over again. And that's why studying just the night before a test is not as great a strategy as reviewing that material little by little throughout a unit and a course. The next stage of neural development is neural pruning. So children's brains have a lot more neurons than adult ones. So at a child's birth, not all the neurons have formed. So this would be an example of how many neurons you have at birth. And then by 7 years of age, you actually have a lot of neurons. So the neurons that don't get used then will be destroyed by a process called apoptosis, which is cell suicide. So the neurons that aren't getting used get pruned away, like you're trimming a tree. And that will allow for the remaining neurons to form lots of synapses with one another, because there'll be room to do so. The term plasticity talks about our ability to alter our connections between axons, and that can happen in a variety of ways. So during neural development, the growth of axons and dendrites, the building of new synapses, which can happen at any point, the elimination of synapses, which also can occur at any point in our life, and then the pruning of dendrites, axon branches, or whole neurons, that happens earlier in our development. And this whole process is called plasticity. So sometimes you may hear that children's brains are plastic. They're not made of plastic. It means that they're flexible and they can alter their connections very easily. And that happens anytime in life, but it's most prevalent before the age of 6, which is why learning a musical instrument or a new language is easier if you could start that before the age of 6. It's not that you can't learn a language or can't learn an instrument, but the neural connections will be a little harder to make as you get older. And also, sometimes, in very rare instances, a procedure called a hemispherectomy may have to happen if there's severe seizures that can't be controlled in any other method. So young children below the age of 6, if they have an entire hemisphere of their brain, which would be completely devastating to most people, if they have it done early in their life, they actually can regain some of their functions that the other side of the brain that's been removed used to perform, because their brains are so plastic. So they'll make new neural connections. So that's plasticity. So why do we have plasticity? It's because our experience that we have shapes our neural connections. So the activities that you participate in, where you go, what you see, everything that you are experiencing shapes your connections in your brain. And plasticity is why and how we form new memories. So we store a memory when we make a neural connection. And it allows for us to be able to reason. That's part of the reason that we are higher order organisms, because we can reason and make those, connections between, if this happens, then this will happen. It is also the key to repairing damage to our brain and our spinal cord. We've talked about neural development. Now we're going to talk about brain development. So the anterior part of the neural tube, so the upper part, will expand to form the brain, which will contain about 86 billion neurons. And the brain acts as the control center. And our major sense organs develop in close proximity to our brain. And that kind of makes sense. They don't have to travel as far to send a signal back and forth. So our eyes, our ears, our nose, and our tongue are all very close to our brain, because the brain controls our ability to see, hear, smell, and taste.